So friends, we were discussing about the factors that affect the protoplast isolation, and we discussed that it is the enzyme mixture that is being used or the source plant material from which we isolate the protoplast. So next factor is the pH and temperature conditions at which we are incubating the plant material along with the enzymatic solution. It has been found that enzymes, they exhibit maximum activity at a pH range of 4.7 to 6 and at a temperature of 40 to 50 degrees Celsius. But it has been found that this high temperature sometimes damage the protoplast. Therefore, it is always advisable to prolong the duration of enzymatic incubation, but at a lower temperature or at a room temperature. There are certain cells which get damaged by light also, therefore such tissues are incubated in dark at room temperature with gentle shaking of around 40 rpm. Now this shaking also helps in the better yield of the protoplast, because this helps in separating the cells as well as better enzymatic contact with the cells. Now tissues can always be cut into smaller portions to increase the surface area for the enzymes. We have already discussed this aspect. Another factor that affects the yield of the protoplast is the type of the osmoticum we are using. As we discussed, that protoplast which we isolate are osmotically quite fragile, and therefore they required almost isotonic solution so that they are maintained. But over the period, it has been found that slightly hypertonic solutions are suitable for the protoplast, or we can say that protoplasts are more suitable in slightly hypertonic solution. One. Second, it has also been observed that slightly hypertonic solution, it provides almost spherical protoplast. Now, why the osmolarity of the osmoticum is important, though we have already discussed, I will just briefly repeat that. Too hypertonic solution will cause lysis of the protoplast, and too hypertonic solution will cause bursting of the protoplast. In order to further stabilize the protoplast, specific stabilizers are also used nowadays, which also helps in maintaining the optimum osmolarity of the osmoticum. And most common stabilizer used is the mannitol, which is used in the concentration of 450 to 800 millimoles. So these were the few factors that govern the or affect the yield of the protoplast or the viability of the protoplast. Now once we have isolated the protoplast, we have checked whether the protoplast, the cell wall has been completely removed from the protoplast or not, and whether the protoplast are viable or not. After doing all these exercises, next step is to fuse the protoplast. If you don't want to fuse the protoplast, we can straightway move on to the culturing of those protoplasts, which we are going to discuss ahead. So coming on to the fusion of the protoplast, which is largely done to produce hybrids. Now protoplast can be fusion of the protoplast can always be produced by broadly two methods. One is spontaneous method and other is the induced method. Now induced is again of three types: mechanical, chemical and electrofusion methods. Now as the name indicates, spontaneous is a method by which protoplasts normally fuse by themselves. Though in nature protoplasts have a tendency to repel each other because of a negative charge present on their surface. But during incubation with the enzymatic solutions, it has been found that certain protoplasts fuse together with the help of plasmodesmata, which are thread-like projections appearing from the cell membrane. Now this kind of fusion is not quite often, and therefore once the enzymatic solution removes the cell wall, they immediately develop plasmodesmata and they undergo fusion. That is why this process we call it as a spontaneous fusion. Next is the induced fusion. As I told you that they have a negative, protoplast have a negative charge on their surface, so therefore they have a tendency to repel each other and do not fuse except in case of spontaneous fusion. Therefore we have to induce the fusion of the protoplast, and this induction of the fusion can be done one by mechanical method and second by reducing the electronegativity on the surface of the protoplast. And this reduction of the electronegativity can be done by using certain chemicals like sodium nitrite, polyethylene glycol along with the high calcium ion concentration, or they can be fused with the help of an electric current, both AC and DC current exposures. So, first coming on to the mechanical induced fusion. Now, this type of fusion is done with the help of an equipment which we call it as a micro manipulator. Now, previously these micro manipulator were largely manual and we have to fuse the two protoplasts under microscope. But nowadays lot of advancements have been made and robotic and automatic manipulators are there which can easily fuse the protoplast under the microscope. So, we just bring the two protoplasts in very close proximity, apply slight pressure onto the two protoplasts causing their fusion. Coming on to the chemical methods of induction of protoplast fusion, and first one is the use of sodium nitrate. Now, this method was first developed by Custer in 1909, but it was explained in detail by Evans and Cocking in 1975, and therefore sometimes people also believe that Evans and Cocking were the person who developed the chemical induced protoplast fusion method. Now, in this hypotonic solution of sodium nitrate is used to fuse the two protoplasts. Now, this hypotonic solution of sodium nitrate causes the swelling of the protoplast and this brings the two protoplasts in close proximity. And sodium nitrate, sodium being positively charged, also helps in reducing the electronegativity on the surface of the protoplast, thereby causing the fusion. But the major problem with this method is the rate of fusion is quite low and the number of hybrids or the viable fusions of the protoplast, because after fusion of the protoplast we obtain hybrids. So, the number of viable hybrids obtained is also very low in case of chemically induced protoplast. Second, it was found in 1973 by Keller and Melcher that if we expose the protoplast towards chemical treatment along with high pH, alkaline pH and high calcium ion concentration, this again helps in improving the overall protoplast fusion and the yield of the protoplasmic hybrids that are obtained. Now, this increased pH and increased calcium treatment was also found to help in development of intra and interspecific hybrids. For this, the protoplasts were incubated in 0.4 molar mannitol having a high calcium ion concentration 50 millimolar and high pH, alkaline pH of 10.5 at 37 degrees Celsius for 30 minutes. And it was found that the fusion and aggregation of protoplast occur within 10 minutes in comparison to the longer durations taken by single sodium nitrate treatment. Second chemical induction method is by treating the cells with polyethylene glycol, and this method was developed in 1974 by Cow and Michailuk, whereas Valen in 1974 during the same year also reported the use of PEG for the fusion of protoplast. And the success of PEG treatment was so that it has now become the most widely used fusogen for fusing the protoplast. Why it has become most widely used? Because it has high fusion frequency, it has low toxicity towards the fused protoplast or the isolated protoplast, and it has high ability of formation of heterokaryons. That is, if you are fusing the protoplast of two different species, then the ability or the yield of those hybrids is quite high in case of PEG in comparison to the sodium nitrate method. With the help of PEG, it was also found that you can fuse plant-plant protoplast, plant-animal protoplast or animal-animal protoplast or even animal yeast protoplasts. It was also found that combining polyethylene glycol treatment along with high calcium ion concentration and alkaline pH improves the efficacy of the method manyfold. there was a significant increase in the overall yield of the protoplast, overall yield of the hybrids that were obtained after fusing the two protoplasts, and moreover the viability of those hybrid was quite high when they were fused, when the protoplasts were fused using PEG along with high calcium and alkaline pH. Now for that, the procedure was protoplasts were treated with 15 to 45% of polyethylene glycol having a molecular weight of 1500 to 6000 for 15 to 30 minutes, and then gradual washing with high calcium ion concentration solution with an alkaline pH was done. Now what happens is, whenever as I told you that the protoplast have a slight negative charge on their surface. Now polyethylene glycol being non-ionic surfactant molecules, they form hydrogen bonds with carbohydrates or certain other polar molecules present in the membrane, and they form a bridge between the protoplasts. So they form a small bridge between the two protoplasts. On one side of the PEG chain they form bond with the one protoplast, and on the other side they form bond with the other protoplast. Now induction of calcium also improves this bridge formation because calcium is a positively charged ion, and this increases the possibility of formation of a bridge between the two protoplasts many folds. That is why induction of calcium enhances the overall yield of the hybrids as well as the viability of the hybrids. Now once this bridge is formed, the two protoplasts are brought closer to each other, forming a dumbbell shaped structure wherein the cell walls are dissolved. Now how these cell walls are dissolved? Because once the bridge is formed, then these are washed with a osmotic solution. Now this causes an osmotic stress on the protoplast, thereby washing off the PEG, but the osmotic stress causes the reorientation of the charge in the plasma membrane of the two protoplasts, thereby causing bursting of the plasma membrane at different points, thereby opening up the joining spaces of the two protoplasts, and thereby leading to formation of a dumbbell shaped structure as you can see in the image. And gradually the two nuclei come together and the dumbbell shape get converted into a spherical shape hybrid molecule. The two nucleus get fused, the genetic material get fused, and in the end we get a hybrid cell or a protoplast. So this is how PEG basically induces the protoplast fusion. Now what are the factors that affect the PEG induced fusion? First one is the physiological condition of the protoplast. As I told you, the young cells, the protoplasts obtained from young cells have more vigor and viability, and therefore the protoplast from the young cells always give better results of the protoplast fusion. Then the density of the protoplast in the fusion mixture is always important. Very less density will obviously decreases the percentage of obtaining the hybrids or chances of the protoplast fusion. It has always been found that 4 to 5% of the protoplast suspension, or 5 into 10 raised to power 4 or 10 raised to power 6 per ml of the suspension concentration of protoplast will give you a good yield of the fused protoplasts. Then handling of the protoplast is another important factor, because once we induce the fusion, too much washing with the enzymatic solution, too much washing with the osmotic solution, too much treatment with the enzymatic solution, or any kind of mechanical stress causes the chances of fusion to decrease. It has also been found that prolonged storage of protoplast before fusion decreases their viability, thereby decreasing their fusion rate. Molecular weight of polyethylene glycol as well as its concentration also plays an important role. As we discussed that the protoplasts were dipped in a PEG solution having a molecular weight of 1500 to 6000, which is normally the preferred molecular weight range for the PEG, because this helps in the formation of the chain between the two protoplasts. Further, 15 to 45% concentration of PEG solution is found to be more suitable. We have discussed the role of calcium ions and alkaline pH in the fusion of the protoplast, as well as the duration and temperature conditions also affect the fusion of the protoplast in the PEG method. Second is the electro-induced method, which was first discussed by Sanda and his group in 1979. In this, wherein in 1982 Zimmermann and Weinken basically described and they first developed the automatic system of the electrofusion of the protoplast. As you can see in this image, there are two electrodes present in the protoplast fusion, and through this two electrodes AC and DC currents are passed, which causes the fusion of the two protoplasts. Now in this whole process, two steps occur. First one is the alignment of the protoplast, and this alignment we always call it as a pearl chain formation or a pearl chain arrangement. So as you can see in this image, between the two electrodes, all the protoplasts are aligned in a chain, like a chain of the pearls, and that is why it is known as pearl chain arrangement or formation. So for this, protoplasts are suspended in a medium of low conductivity having mannitol and calcium chloride, and this low conductivity solution helps in stabilizing the plasma membrane. So in order to avoid any kind of osmotical stress, initially the protoplasts are stabilized in a low conductivity solution, and then they are exposed to a non-homogeneous, low voltage and high frequency AC current for 30 to 60 seconds. Now this AC current creates a dipole charge in the protoplast. So we, in simple words, we can say protoplast become dipole, and this process we call it as protoplast dielectrophoresis. So opposite charge are formed on the two areas of the protoplast. Now once the protoplast become dipole, they tend to get aligned themselves in the fusion mixture, thereby creating a pearl chain arrangement. So fusion mixture, we know that it contains non-ionic osmoticum, usually 0.5 molar mannitol, and along with calcium ion concentration, higher calcium ion concentration and alkaline pH, and whenever this AC current is passed, they become dipole and they align themselves in a pearl chain formation. As you can see in the first image on the left, the protoplast, one is chlorophyllated and other is non-chlorophyllated. So these are randomly arranged, but after passing the AC current, they align themselves in a straight chain, which we call it as a pearl chain formation. In the second step, the fusion of these aligned protoplasts occur, and to fuse these protoplasts, we pass a small pulse of high voltage DC current, and these pulses are for 40 to 40 microseconds, 4 to 40 microseconds of the DC current. Now with the passage of DC current, it causes a irreversible breakdown of the plasma membrane, and the two protoplasts, they start getting fused at different points, leading to the fusion of the cellular contents. And once the cellular contents are fused, the cell or the fused protoplast, they get converted into a spherical shape. So this is how the electrofusion method fuses the protoplast. Now certain advancements were made and a microelectrofusion method was developed by Knopp, wherein the two protoplasts were placed in a droplet of the mannitol solution, and then small wire shaped electrodes were put into those drops. An electric current was passed and the two protoplasts were fused under the microscope. Therefore the method was known as microelectrofusion. Now for a successful electrofusion method, good quality of protoplast should always be used. The strength and duration of AC current exposure should be optimized, because too prolonged exposure of AC current or too high voltage AC current will cause lysis of the protoplast. Likewise, strength and pulses, intensity of the pulses as well as duration of the DC current pulses should always be optimized, because it varies from the material to material or the plant material to plant material. Now once we fuse these protoplasts, or if you haven't fused, you isolated the protoplast, the next step is to perform the protoplast culture. Now, in order to initiate the protoplast culture, appropriate density of the protoplast culture should be there, and this density can always be determined by using hemocytometer. And the optimum density we have discovered 5 into 10 raised by 4 to into 10 raised by 6 per ml of the suspension, which is good for fusion as well as for the protoplast culture. So, once you have optimized the protoplast density, then we establish the culture. Now, the culture of the protoplast can be established either by in the form of callus cultures, because we allow these fused protoplast or the isolated protoplast, allow them to develop their cell wall, and once the cell wall is developed, they start dividing onto the artificial media. We can also develop protoplast culture in suspension, which we call it as suspension protoplast culture, or another method is double layer method, wherein one layer is of solidified media and above it is the liquid media, and another one is the agarose droplet or bead method. So, coming on to the solid or semi-solid media method or the callus method, wherein agar, agarose or alginate are used as the gelling agent and the protoplast are suspending in double the density than the required. Media and gelling agents are again prepared in twice the final concentration, and then the protoplast are suspended in the mixture before it gets before it gels, and then the suspension is transferred into the petri dish where it is allowed to cool. Once the media gets solidified, the protoplast media can be cut into blocks and then it is transferred onto the liquid media for further growth. So, this is how the solid culturing of protoplast is done. Next is the protoplast culture in the liquid media. Again, in this protoplast are suspended in the liquid culture. Now, these protoplasts can be simple single isolated or the fused protoplast. So, these are suspended in the liquid culture media at the appropriate density into the culture dishes as or in the form of micro droplets into those dishes, and then dishes are sealed with the gas permeable membranes in order for the proper gaseous exchange. There are certain advantages of liquid culturing of protoplast, because some of the species, it has been seen that protoplast of some of the species would not divide on a callus on a solid media. Further, osmotic pressure of the media can be effectively controlled in the liquid form. Density of the cells can always be reduced in the liquid form by adding the fresh media. We have already discussed in the suspension culture lecture. Cells of the special interest can be isolated after culturing them from the liquid suspension, which is more easier in comparison to that of the solid medium. Medium can be changed from time to time, and the liquid media also permits rapid diffusion of the nutrients as well as gaseous exchange in comparison to the solid media. So, these were the few advantages of culturing protoplast into the liquid media. Third method is the double layer culturing. As I told you, in this, a layer of semi-solid media is dispensed in the bottom of the petri dish and it is allowed to get solidified. On this solid media layer, same volume of liquid media containing protoplast in twice the concentration is poured over the solid layer. Normally, a layer of filter paper or a membrane is put between the liquid and the solid media, between the two phases, which helps in stimulating the cell wall regeneration and helps in sustaining the mitotic divisions of the cells. Fourth method of protoplast culturing is the agarose droplet bead method, wherein the isolated protoplast are cultured in a drops of the culture media, and these drops are formed with a volume of approximately 50 microliter of the media, and these drops normally hang from the lids of the petri dishes. Now, the protoplast normally, since they are heavy, they sink to the meniscus of the droplets, and once they sink to the meniscus, this helps in the adequate exchange of gases between the droplets and the atmosphere, or we can say it leads to the adequate aeration of the protoplast. Now, this kind of method is found suitable for the protoplast at low densities and for evaluating the composition of a range of culture media. Though the method is time consuming and droplets are tedious to prepare, this method is widely used in on experimental basis to study the effect of the components of the culture media. Now we will discuss the factors that affect the culture growth of the protoplast. First one is the formation of cell walls. Now, in order to for the growth of the protoplast cultures, the protoplast must develop the cell wall, because until they develop the cell wall, they will not undergo the cell division or they will grow. So, cell wall formation is an important factor for the proper culturing growth of the protoplast. Then the source of the protoplast, whether the protoplast has been obtained from young plant material or the old, young ones have better viability, whether the protoplast were developed or the cells from which protoplast have been isolated were developed from under aseptic conditions or not. All these factors, they basically help in the proper maintenance of the growth of the culture. Protoplast with lower viability, obviously the growth will be stunted. Protoplast with better viability will have a better growth. Then the plating density of the protoplast. We discussed that lower density will have lesser growth, higher density, too much higher density or the too much higher number of protoplast per ml of culture media will again bring in the competition for the nutrients, again the growth will be stunted. So, optimum density of the protoplast should be there. Then culture media, the composition of the culture media should be appropriate, which supports the rapid cell wall development, which supports the rapid divisions of the cell and the culture growth. Ammonium ions have been found to be toxic for the protoplast of many species, and therefore they are normally avoided in the culture media meant for the protoplast culture. The osmolarity of the osmoticum is again an important factor that helps in defining the growth of the protoplast culture. Now, it has been found that after 4 to 5 days of the culture, osmolarity is gradually lowered by periodic addition of the few drops of fresh media, which lacks the osmoticum until multinucleated colonies are formed. So, gradually we have to decrease the osmolarity in the culture media, but this again should be gradual, as sudden decrease in osmolarity will cause damage to the protoplast. Various agents like mannitol, sucrose and glucose are used to maintain are used as a stabilizer to maintain the osmolarity of the media. There are certain physical factors like electroporation is done, which increases the cell division, which enhances the cell wall regeneration rate, as well as it enhances the plating efficiency. Treatment with higher temperatures, which we call it as heat shock or cold treatment, also helps in improving the cell regeneration efficiency as well as the plating efficiency. Protoplasts which are normally light sensitive should always be protected from light and should be developed under dark conditions. Optimum room temperature, optimum temperature of the media should be maintained. So, all these physical factors also have an impact on the growth of the culture. Now, we will discuss about the somatic hybridization. So, as I told you that once the protoplast fuse, we call this process as somatic hybridization, and there are three major aspects of somatic hybridization, that is fusion of the protoplast, then formation or the selection of the hybrid cells from the fusion suspension or the fusion mixture, and then characterization of those somatic hybrids. Now, coming on to the first aspect, selection of hybrid cells. Fusion we have already discussed, then selection of hybrid cells. It has been found that whenever the protoplasts are fused with electrofusion method, there is higher percentage of proper hybrid cells formed in comparison to the chemical fusion method. Though I have already discussed this, electrofusion method is always considered better than the chemical fusion method, and higher yield of hybrid cells can be obtained from the electrofusion method. Another reason why chemical fusion has lower yield, because in chemical fusion the fusion mixture that is formed is quite heterogeneous. In that mixture we have homokaryons, that is when the protoplast of the same cells from the same species are fused. We also have heterokaryons when the protoplasts of two different species are fused. Parental type protoplasts are also there, and other combinations of cytoplasmic combinations are there, that is mitochondrial genome from one to plastid genome from other, or nuclear genome from one, nuclear genome from other. So such kind of multiple combinations or heterogeneity is there in chemical fusion, and therefore selection of hybrid cells from such chemical fusion method is quite difficult, and the frequency of getting desirable fusion product is quite low in chemical type than in the electrofusion type. So friends, in the next lecture we are going to discuss that how you are going to identify these hybrid cells, what are different hybridization that occur when we fuse the protoplast, and what is the application of different types of hybrids that are formed during the protoplast fusion. Thank you.